Spiegel. Welcome to the first full day of the World Stem Cell Summit here in Miami, Florida. I am very, very pleased to be able to welcome the 6th Annual Japan Symposium to our meeting. Japan is playing a critical part of the regenerative medicine and stem cell research world. And here we have a combination of government leaders, industry, and academia to present the narrative that you need to succeed, because you will not understand this field and what's going on unless you understand what's going on in Japan. I'm very honored to introduce our chair of this session, my esteemed friend, Dr. Takashi Asada. Thank you.
Thank you. Hello, everyone. My name is Takashi Asada from Kyoto University. It's a very good time for this symposium for 6 times. We are very pleased for this symposium to this big auditorium. And also, there are many persons from Japan to attend this time. And I already told Professor Okano from Keio University, he cannot attend this time, but he can talk about by web for the second speaker. And first speaker is Norio Nakatsuji from Kyoto University. He'll talk about overview of this symposium and also his company work. So please, Norio.
Thank you very much for attending to our symposium from this early morning session. So I'd like to give you a very brief overview and few words about activity of my startup company originated from our work at Kyoto University. So I told you last evening, but I believe the key to success with such a very complex long-term effort for the regenerative medicine and new therapy, the key to success is multidisciplinary technology, R&D, and multi-sector collaboration. And I have been working for that in Japan to that purpose. And today's session is some showcases, best example of the people working at academia and government and industries from Japan. And actually, I have been reading for almost 10 years the Japanese government supported project of collaboration between academia and industry. Japanese government divided into several ministries, and this part is, my project is supported by METI, Ministry of Economy, Trade and Industry. So focusing on the commercialization and industrialization. And I was very lucky to assemble the very best players of the industries, companies and academia from 2010, including Professor Okano of Keio University. And current project still going on is a project with the big player of the industry, Fujifilm, you know very well, I think. So it's purchasing CDI for several years ago. So this part currently is a very comprehensive project of starting from the new culture system of low-cost, high-quality culture system to the differentiation by, for example, using only small chemical compounds and 3-dimensional construction of the tissue-like structure and quality control and freezing, and to the end is transportation of the cell materials of to even the international travel by keeping the temperature constant. Something so very comprehensive project mainly driven by each companies with good technologies. So my talk is to the three-dimensional construction of the micro tissue. So this is a Kyoto University work. Rather than currently use the two-dimensional adhesion culture or the single cells, we try to make low-cost, quick method to make three-dimensional multicellular structure using the cardiomyocyte as the first example. So this is some view of our microtissue with using aligned nanofibers. Actually, nanofiber is a rather thick nanofiber, rather microfiber. And with a bundle of aligned nanofiber, it's easy to be produced by electrospinning. And then put our cardiomyocyte made by pluripotent stem cells using only low molecular weight chemical compounds make nicely aligned structure resemblance of the actual heart tissue. So rather than making not only cardiomyocyte, but making a small piece of heart muscle structures. So the result is there's a very nice devices you can take up by forceps to be used for drug testing, or maybe could be future can be used for cell therapy using biodegradable nanofibers. So this technology developed at Kyoto University of ISEMS, where I am the founding director. That's a patent license to our startup company, Stem Cell and Device Laboratory, called SCAD. Then I don't tell the detail, but you could look at the website. So it's a very young, but still already going to the 4th year, and with recently secured financing Series B and several $1 million. And so now starting to sell our product. Our product is named SCAD-MT, is a microtissue cardiomyocyte, and we plan to expand the line of product. So it looks like this one. We actually, first we made this 10 millimeter square-like structure. This should be smaller for the drug toxicology assays as the first purpose. So we are now process of making smaller, more handling easy way for the high throughput, even high throughput screening. But I can't tell today the detail of that, but soon available. And this is this is a three-dimensional view, so difficult to take good pictures, but this shows nicely aligned fiber and cardiomyocyte and the sarcomeres, so like the heart tissue. And putting the cardiomyocyte from the, in this case, from iPS cell lines to make three-dimensional tissue-like structure, enhance maturation. For example, this is a cardiomyocyte-specific gene expression enhanced to even closer to the adult level rather than fetal level. And stability for if you know about the toxicology assay, one currently frequently used method is multi-electrode array system, and we could pick up the very stable signals from every electrode point to test the toxicology. So nice signals and stability of device to device. So this is 14 devices and get the similar signals. So, and toxicology could be drug response is reliable as expected using a model chemical compound and with other compounds. So this is a current summary of our product originated from the Kyoto University academic research result. So rather than currently widely used single cells of cardiomyocyte from pluripotent stem cells, we are making micro tissue-like cell devices. We are almost successful to make smaller devices, and that shows more maturity and reliability, stability. So we plan to actually expand this beyond the cardiomyocyte. I cannot tell the detail, but there are obvious candidates of the important cell types for the drug testing. And if we find some partner for clinical application, we could use such a three-dimensional tissue for the cell therapy, tissue therapy aspect using biodegradable polymers for making fibers. Okay. Thank you very much for your attention. So following is the best example of the Kyoto, Japan scientist and industry. Thank you very much.
Okay, it's my great pleasure that I can make a talk in this great opportunities. Okay, so my name is Hideyuki Okano. Okay, thank you very much for kindly inviting me such a great opportunity. I'd like to thank Professor Nakatsuji and Asada who kindly invite me. Okay, unfortunately, I cannot be at Miami, but I'll do my best from Tokyo. Okay, today I'm going to talk about the challenge toward iPS-based cell therapy and modern CNS disorders. Okay, so as you know, it has been long believed that adult mammalian central nervous system does not regenerate upon injuries, as Dr. Santiago Ramon y Cajal had indicated long time ago. But so he described in his book that so once the development was ended, the forms of growth and regeneration dried up irrevocably. This is so-called Cajal's dogmas. But we wanted to challenge for this dogma. Okay, by taking advantage of stem cell technology and other techniques. The regeneration of the central nervous system does indicate regrowth of disrupted neural axons, and so replacement of lost neural cells and recovery of lost neural functions. These are very important aspects of CNS regeneration. So we call the 3 R's of the CNS regenerations. Okay, so the question is that how can the regeneration of the central nervous system be achieved? I believe it's important to recapitulate at least some aspect of the normal CNS development. Okay, so but notably the development of the central nervous system is initiated by the induction of the neural stem cell or neural stem-like cells. So it'd be very reasonable to activate or manipulate neural stem cell to recapitulate normal CNS development for the regenerating damaged central nervous systems. The neural stem cells are multipotent stem cells that present with the central nervous systems that has both multipotency and so self-linear activities. the single neural stem cell capable of generating various types of cells within central nervous system, including neuron, astrocyte, and oligodendrocyte. This is multi-lineage potency, and then neural stem cell can be expanded at self-linear state. This is also self-linear abilities. Neural stem cells are present in the fetal brains abundantly that are expandable in vitro. They are present in the adult brains only at very small levels, but they are not easy to expand. Okay, so by using such a characteristic of the neural stem cells, we plan the stem cell therapy for the spinal cord injuries. The traumatic spinal cord injuries, okay, that affect many people, including young people, and can result in severe damage, and so the leading to paraplegia, tetraplegia worse. But so we wish to challenge toward the regeneration of the injured spinal cord. Okay, so by using these stem cells. But we have to realize that the pathophysiology of the spinal cord injury changes according to the time progressions after the primary and mechanical traumas. Spinal cord injuries begins with primary mechanical traumas and caused by the car accident, sports accident, and accidental fall, etc. It's for the series of events, such as inflammations, and this so-called secondary injuries. Actually, so these the inflammatory process continues at least several days after the primary mechanical trauma, called acute phase. Okay, so but after several days after the injuries, the inflammation is downregulated. It's followed by the delayed event, so we call it as secondary injuries. So-called, subacute phase. Subacute phase is include both regenerative event and the degenerative event. So if the regenerative event is the strongest, the patient recovers spontaneously. But so the degenerative event is strongest. The prognosis of patient is very poor. Okay, so after 6 months after the injuries, the state of the patient situation becomes more fixed. So we call the chronic phase. And the chronic phase is characterized by severe axonal degeneration, scar and cyst formation, and permanent loss of spinal functions. So the question is when is the most appropriate time point for the stem cell transplantations? Actually, if you transplant, the acute phase, so the viability is so poor. And if you transplant chronic phase, the strong scar formation prevent the therapeutic effect of the stem cell transplantations. Okay, so and then we found that stem cell transplantation gives the maximum therapeutic output when they are transplanted, so within several weeks after the injuries. Okay, so based on these, before the born of the iPSA technologies, we plan to develop the neural stem cell transplantation for the spinal cord injuries. Okay, so first we transplanted rat fetal neural stem cell for the spinal cord injury in the adult rat in 2002, and then we planned human fetal neural stem cell transplantation with the spinal cord injuries. But there, as you notice, there's a strong gap, a big gap between them. So in order to fill this gap, we have performed human fetal neural stem cell transplantation for the spinal cord injury in non-human primate models. Actually, in 2005, we have a reported paper about development of the contusive spinal cord injury in non-human primate using the small monkeys called common marmoset. We induce the contusive spinal cord injury and in marmoset, and so at the level of C5. And then we transplant human neural stem cells and could induce functional recoveries. So these findings was reported not only by the original paper, but also various media, like the Washington Post. So we believe these are the big step towards the clinical trial of the neural stem cell transplantation for the patient with spinal cord injury. But so various ethical problems prevented our clinical translations. Okay, actually, so in 2006, Japanese guideline on the use of human stem cell for the clinical research was released. And believe it or not, so this guideline did not allow us to use aborted fetus derived cells and even the human ES cells. This is very surprising and also shocking to us. Okay, so then in order to develop the stem cell therapy, we need a big breakthroughs. How to prepare the neural stem cells. So in the same years, the guideline was released. Okay, so the groundbreaking paper from the Shinya Yamanaka's papers appeared, as you know, the iPS cell technologies. So adult somatic cells, like the skin fibroblasts, can be reprogrammed into the ESL-like cells, named induced pluripotent stem cells, iPS cells. Okay, so the scheme is that so the fibroblasts can be induced in the iPS cells, and then we developed the method to induce the neural stem cells. Okay, neuralized embryo body with lower amount of retinoic acid or as noggins, and we could induce the neural stem cell by FGF2 treatment. Okay, so these neurons, iPS-derived neural stem cell, differentiated into the neuron, astrocyte, and oligodendrocyte. Okay, so these neural stem cell transplanted into the mouse spinal cord injury models. So I believe so the movie is not perfectly transferred from Tokyo to Miami, but let me show you. Okay, so this is a contusive injury at level TH10. Okay, so without transplantations. But if you transplant iPS-derived neural stem cell, okay, so the subacute phase, it shows that nicely the function recoveries. So coordinated, this slide shows that coordinated movement both between the forelimb and hind limb. And animal can stand up with their hind limbs, indicating the functional recovery of the spinal cord. Okay, so then we also noticed that transplanted iPS-derived neural stem cells are differentiated into the oligodendrocyte, and so and they have nicely myelinated, demyelinated axons. Okay, so and also 2007, Yamanaka's group and developed human iPS cells, and this was used for the also used to induce neural stem cells like this method. Okay, so then these iPS-derived neural stem cells are transplanted, so endodyskid, spinal cord injury models. And transplanted iPS-derived neural stem cell nicely differentiated into the neurons. They were shown to make synapse with host neurons. So they have relate disrupted neural, disrupted synaptic connections, like this. Okay, so the transplantation of the human iPS-derived neural stem cell promoted functional recovery after spinal cord injuries. Okay, then we have examined transplantation into non-human primate models. This is control. Okay, so contusive injuries. So animals show the paralysis both forelimb and hind limbs. But if you transplant, iPS-derived neural stem cells, so it shows a nice functional recoveries. Both the function and the forelimb and hind limb recovers. And this is quantitative data. So significant functional recovery could be observed by transplantation of the iPS-derived neural stem cells. Okay, so then we have found that the number of the myelinated fibers are increased by transplantation of the iPS-derived neural stem cells. Okay, so based on these, we believe these are possible mechanisms of functional recovery by the iPS-derived neural stem cell transplantations. The one mechanism is that trophic actions. So iPS-derived neural stem cell produce various trophic factors, and they might have induced various endogenous and regenerative pathways. Okay, so iPS-derived neural cells might have induced the regeneration of the host neural axons, And also, they might have recruited endogenous neural stem cells. So this is so-called trophic actions. In addition to these non-cell autonomous mechanisms, iPSDR neural stem cells should have myelinated demyelinated axons like this. And also iPSDR neural stem cells are differentiated into neurons and make synapse, and so are reconstructed the neural circuit by the synaptic relay. So both trophic action and such a cellular replacement mechanism are very important mechanisms for functional recoveries. So taken together, so by using mouse and human iPS cells, they are induced in neural stem cells and transplanted into mouse and non-human primate spinal injury models. And so we found that we could observe long-term functional recovery by transplantation to neural stem cells, without any tumor formation, if we choose appropriate cell lines. Okay, so if we consider about the clinical translations, so this is an autograft model. So patient-derived somatic cells induced in the neural iPS cells, neural stem cell transplant to the patient. But if the neural stem cell were to be transplanted within 1 month after the injuries, this scheme becomes very difficult. Okay, so because it takes 3 months to establish iPS cells and 3 months to induce neural differentiations, and 1 year for the quality control, including tumorigenesis. So that means that we need iPS cell in advance. And then we can start a new collaboration with Cyrus. So we have to establish a safe iPS cell stock for the clinical translation from HA homozygous superdonors. And then Keio, we prepare the clinical grade iPS-derived neural stem cell stocks. And we examine the neural differentiation and expansion of the iPS-derived neural stem cell at the GMP levels. And we do the quality control and safety issues of clinical grade iPS-derived neural stem cells. Okay, these are the possible procedures of the clinical grade of the iPS-derived neural stem cells. We examine 3 methods. Protocol A, so prolonged embryo body formation, SFEB, and the neurosphere formations. And protocol B, embryo body formation, and rosette, and also neurosphere formations. And protocol C is embryo body and rosette and sphere, and then monolayer again. So we have examined transplantation of neural stem cells as prepared by 3 different methods into the brain of the nog mouse, 1 million cell per striatum. And also we performed the transplantation into the injured spinal cord enodiscid mouse. And we have performed histological evaluation about 12 to 26 weeks after the injections. Okay, so these are the results. Okay, by the protocol A, so these cells had no tumor genicity and no abnormal cells. But protocol B and C, and some extra medullary outgrowths was observed, even if they are not tumors by the histologies. But protocol C, we noticed that some ossifications. Possibly this is contributed that neural crest-like cells, so included in these neural stem cell neurosphere populations. And so all the historical data now are available in the database from this website, from this URL. Okay, so then based on these, we have chosen protocol A to prepare the clinical grade iPS cells. We also developed a fail-safe system to prevent tumor genicity by 2 methods. And one is gamma secretase inhibitor pretreatment before the graft of the iPS-derived neural stem cells. Okay, so the other is the introduction of the ICASPase-9 system in iPS cells. Okay, so by the gamma secretase inhibitor treatment, 1 day before the transplantations, and this treatment had a long-term effect. Okay, so grafted cells are differentiated into more mature neurons and prevented tumor-like outgrowths and maintained the greater functional recovery. And so let's improve the safety of the iPS-based therapies. Actually, we have incorporated such a gamma secretase inhibitor treatment in your clinical protocols. And also we have developed the procedure for the production of the clinical grade iPS-derived neural stem cell. Okay, so by using iPS cell stock from Cyrus, we expand the iPS cell and make 2 passages to make the working cell stock. Okay, so these will then go by using the protocol A, we produce iPSDM neural stem cells. But several passages, we make the final product. For the final product, we examine sterility assay and toxin, mycoplasma, and RNA-seq, and DNA-seq, and flow cytometer, and so neurosphere formation, and tumor density test, and also examine functional recovery. Okay, so then we finalize the clinical grade of the iPSDM neural stem cells. So this is the roadmap, and so the SIRA is going to establish clinical grade iPS cells. And so in Keio, we do perform the neural differentiation and expansion of the optimized method. And so the preparation of the clinical grade iPS cells. Now we submitted a protocol to the IRB of the Keio University. And so we wish to start the first human trial in this year. Okay, so further indications of transplantation, iPSDM neural stem cell would include the chronic phase spinal cord injuries, and also stroke, and so multiple sclerosis, and Huntington disease, and other neurodegenerative disorders. Okay, so then how can we challenge for the spinal cord injury for the chronic phase? Okay, so neural stem cells should be combined with the treadmill exercise for the chronic phase. So by transplantation, the iPS cell by itself, it do not show the functional recoveries by itself. But so we have combined them into rehabilitations of the by treadmill training, like this. We do this kind of training every day. It resulted in as significant functional recovery. So the idea is that when we transplant the iPSDM neural stem cell in the subacute phase, so it causes remyelination, regenerative fibers, and increase in the central pattern generators. But so if we use transplant in the chronic phase, the only result partial myelination, some regenerative fibers, increase the CPG activities. So only limited effect. But if you perform treadmill training, so microenvironment in the subacute phase, continues a prolonged time. Okay, so so this means that the treadmill training shows causes changes in microenvironment suitable for transplantation neural stem cells. For the clinical applications, combined therapy as iPS therapy and also cyborg type robot, how will be carried out. This will be done with the collaboration with the Cyberdyne and the Keio universities. This is Cyberdyne, how we do the treadmill training. So very weak current on the muscle will be amplified, and so it will be translated into the leg movement. Okay, so now we are doing the clinical trial of the heart by itself, and it will be combined with the iPSDM neural stem cell therapy in the very near futures. In the remaining a few minutes, I'd like to also talk about another side of the iPS cell technologies. So investigation neurological and psychiatric disease model, diseases using iPS cell technology for developing disease-modifying therapies. Okay, so so we can easily generate iPSDM neural stem cell, patient-derived iPS cells, and they can be induced for the disease-sensitive cells. For example, motor neuron in the case of the ALS. These ALS motor neurons can be compared with the healthy donor-derived motor neurons. We can then perform the disease modeling, drug screening, and toxicity test. Okay, so and so we can introduce for example, ALS-related mutation into the healthy iPS cells by genome editing. When they induce the motor neuron, if we can recapture disease-like phenotypes, we can tell the causal relationship between genotype and phenotype. And that can be tested in a wide variety of cells by taking advantage of the prepotency neural iPS cells. Ashe group established iPS cell lines from about 40 neurological and psychiatric disorders. These blue color indicate the pediatric neural disorders, including Rett syndrome and neurodevelopmental disorders. So cerebral cortex developmental defect and leukoencephaly, the dysmyelinations, again, Pacemaker disease. These are hereditary dysfunction of the sensory organs and mental disorders. These right panels can indicate late onset neurological disorders. Particularly neurodegenerative disorders like Parkinson's disease, or ALS, or Alzheimer's disease, or multiple system atrophies. So by using iPS-based drug screening, we start a new trial for the Pendret syndrome that shows the progressive hearing loss and also the ALS, devastating motor neuron diseases pretty soon. So in addition to such application as regenerative medicine or disease modeling and the drug screening or the cancer immunotherapies, lots of indication has been suggested for the iPS technology. So in vitro organogenesis, human embryology, and also human-animal chimera, and so and to produce, for example, pancreatic organ within the pig, human pancreatic organ within the pig embryos, or the human reproductive biology, and investigations, evolutions. So epigenetics during reprograms. Many exciting projects are there by using iPS technologies. So so much for data, and so these are collaborators. We have collaborated seniors since 2006. We have performed a long-term collaboration with Masayo Nakamura, professor of orthopedic surgery in Keio. And also, we also collaborate with Dr. Masashi Aoki's iPS-based drug screening for the ALS. And these are patient advocate and always encourage us. Thank you very much for your attention. That's it. Thank you. Thank you very much for attention.
Web discussion will be closed. And then next speaker is Yoji Sato. He's the head division of cell-based cells products and National Institute of Health Sciences. He talked about Japan regulations and also other recent progress of his research and also many kind of things. He'll talk about that. Dr. Sato, please.
Thank you, Takashi. Good morning, everyone. First of all, I'd like to thank Takashi and Bernie and the organizers to invite me to this wonderful symposium. Today, I would like to talk about current activities for efficient translation of cell therapy product. More specifically, I'd like to introduce the nationwide regenerative medicine data system in Japan and multi-site study for validation of test method, particularly for tumor genetic assessment of cell therapy product. So let me start with the nationwide data system. This slide shows you the regulation for regenerative medicine in Japan and the United States. As you can see, there are remarkable differences between the two countries. And regenerative medicine, regenerative medical product or cell therapy product with more than minimum manipulation in the shadow areas in the slides are required to be authorized by regulatory authorities before marketing and clinical use. So in Japan, a commercial distribution of cell therapy product is regulated by Pharmaceutical and Medical Devices Act, PMD Act for short. In contrast, non-commercial use of cell therapy product in hospitals or clinics is regulated by Act on the Safety of Regenerative Medicine, ASRM for short, which provides distinct review process. In the United States, as you can see, a common regulation applies regardless of the purposes. In conventional process, sponsors need to confirm both efficacy and safety of their products for marketing authorization. As you may have already known, in Japan's PMD Act, we recently introduced a step of conditional and term-limited approval, which accommodates early practical application of regenerative medicine products or cell therapy products. So if clinical data are likely to predict efficacy and confirming safety, the product may be eligible for the conditional and time-limited approval. A problem in Japan is that the quality of data from non-commercial clinical researches have not been necessarily compliant with good clinical practices under the PMD Act. So the data cannot be used directly for marketing authorization. This is our challenge in Japan. In addition, the conditional and term-limited approval, we need to further confirm the safety and efficacy during the post-marketing study. So collection and analysis of the real-world data are very critical and another challenge. So 2 years ago, the Japanese Society for Regenerative Medicine formed a nationwide open innovation platform to promote clinical researches, which is called a National Regenerative Medicine Consortium. The consortium consists of 5 functions, namely a consultation for clinical researches, development of human resources, development of clinical research database, data system, coordination of academia industry translation, and interactive communication with patients and citizens. So let me briefly introduce the data system. It is called NRMD, which stands for National Regenerative Medicine Database. So NRMD is for both regenerative medicine clinical researches and approved cell therapy products, and is also compliant with good post-marketing practice. So in other words, it's computerized system validation compliant. Computer system validation is a documented process of assuring that a computerized system does exactly what it's designed to do in a consistent and reproducive manner. So NRMD consists of systems for non-commercial and commercial clinical researches and for post-marketing studies, which are called NRMD-CR and NRMD-PMS, respectively. So we are building them in collaboration with relevant academic societies and Japanese regulatory authorities, PMDA. So NRMD also has a database and an archive of the cells actually used for the patients. So we expect that this database enables a seamless translation or reverse translation of researches from clinical investigation to PMS by acquiring real-world data of all clinical cases into a system with common data quality assurance. So, for example, data obtained from non-commercial researches under the ESRM might be used as historical controls for PMS of new products under the support of the academic society and the regulatory authority. So first in human clinical researches for an allogeneic iPS cell-derived products is expected to be the first case on NRMD, and more specifically, allogeneic iPS cell-derived retinal pigment epithelial cells for patients with age-related macular degeneration. So let me move on to the multi-site study for validation of testing method. So So human pluripotent stem cells, PSC, have the potential to revolutionize regenerative medicine, as you know. But clinical trials on pluripotent stem cell-derived products will start soon in many countries. However, undifferentiated pluripotent stem cells and cell transformed during the manufacturing process may form tumors in patients. That's a problem. So tumor formation is an independent risk for cell therapy products, especially for pluripotent stem cell-derived products. So tumor genetic assessment of cell therapy products is one of new challenges for sponsors and regulators. So, however, there's no internationally accepted consensus for the quality and non-clinical safety evaluation of cell therapy products, including tumor genetic assessment, biodistribution, as well as biocompatibility. So let me explain two abbreviations. Pharma concept is a name of a committee, which stands for Committee for Non-Clinical Safety Evaluation of Pluripotent Stem Cell-Derived Product. I think, cell-derived therapeutic products or something, not product T, the Forum for Innovative Regenerative Medicine. And a measure is a name of study, which stands for Multi-Site Evaluation Study on Analytical Method for Non-Clinical Safety Assessment of Human-Derived Regenerative Medical Products. So the mission of Measure project is to provide sound science-based and globally acceptable consensus for safety evaluation policy and the R&D of pluripotent stem cell-derived products, especially validated method for tumor genetic evaluation, which are in alignment with regulatory direction and international standard, will be developed through multi-institutional joint researches. The above is conducted through Measure project, which is an AMED-funded research project of Pharma concept and NIHS, my institute. So there are a lot of testing methods which are published in scientific papers. So we are trying to validate some of them. So out of them, the consortium is currently validating 6 test methods, including 1 in vivo tumor genetic testing, the 4 in vitro testing for detection of residual iPS or ES cells, or detection for detection of malignant transformed cells, as well as 1 in vitro biodistribution testing. So the preliminary data around the standard protocol will be available in the end of next March soon, and the following March, respectively. So the project is conducted by NIHS, my institute, and 6 pharmaceutical or associated companies in collaboration with some other pharma concept companies, as well as CROs. So we keep discussing the quality and safety assessment of cell therapy products with international academic societies like HESI or IABS, ISCT, and ISCI, and Japanese regulatory authorities such as MHLW and PMDA, as well as industry associations, including Pharma. So our main platform of the international discussions is a committee of HESI, Health and Environmental Sciences Institute. So this slide shows you the members of the Committee of Cell Therapy Tracking, Circulation, and Safety of HESI, CT tracks for short. So we are currently discussing test method for the safety and quality of cell therapy products, including tumor genetic assessment and biodistribution. So we have a poster presentation over there. So if you are interested in it, please stop by this afternoon. So cell therapy products or regenerative medical product need to have efficacy and safety, of course, and quality for ensuring the safety and efficacy, and specifications for ensuring the quality. But without knowing characteristics of the test method, neither sponsors nor regulators can justify the efficacy, safety, quality, and specifications at advanced regenerative medical products or cell therapy products on sound scientific basis. So I would say no testing method, no product. So in any way, we hope that our activities contribute to fostering consensus or global consensus on safety evaluation policy in the development of cell therapy products. Thank you for your attention.
Thank you, Dr. Sato. From now, next speaker from industry keynote speaker. He is Akihiko Isai from Vice President Astellas Pharma, Inc., and also Vice Chairman of Forum for Innovation-Related Medicine. It's called Pharma. Please, Dr. Isai.
Thank you, Dr. Asada and Nakatsuji. I'd like to say a lot of thanks to the WSCS organizers to invite me this very interesting meeting. Anyway, so good morning, everyone. So my name is Akihiko Isai, and I come from Astellas Pharma of Tokyo in Japan. And so I am also a vice chairman of Japan Forum for Innovative Regenerative Medicine, so called Pharma, which is Japanese Industrial Association of Regenerative Medicine. So the name of Pharma is also shown in my previous presentators. Okay, so these are today's contents. In my presentation, so I will address how Japanese pharmaceutical companies are engaged in the regenerative medicine, taking Astellas Pharma as an example. First, I am going to explain business models of regenerative medicine for pharma. The second, so I touch upon the concrete activity of regenerative medicine in Astellas. The third, I am going to introduce a pharma. Okay, the first subject. Anyway, so this is a future market size of regenerative medicine estimated by Japan Ministry of Economy, Trade, and Industry, so-called METI. The market size of regenerative medicine is so rapidly increasing. Global market size will be around $380 billion in 2050, very large. And Japan takes around 80% share of the global market. And you know the number one market share is being taken by the United States, needless to say. To enter this attractive business, what does Japanese pharma consider? As you know, regenerative medicine requires cutting-edge science, new technology, novel business expertise. To respond to this new field, pharmaceutical companies have to change their own authentic business value chain and create a new plan to enter this business. So in making plan to enter this business and establish business archetype, firm should consider the following components. They are first, cell type. What cell type we are going to use? And cell process technology, the expected effect, and so the delivery, indication and diseases, and treatment mechanism, and regulatory body and regions, and reimbursement. Taking all of them into consideration, Pharma can plan its own business models and business plans. Anyways, I'm sorry, this is a very busy slide. And so it explains kinds of value chains in pharmaceutical company. You know, so one of the strengths of Pharma is that Pharma traditionally has most value chains to construct healthcare business, which includes research and development, manufacturing, delivery and logistics, sales and market affairs and access, and interaction of regulatory and reimbursement bodies. So Pharma has all the value chain inside. However, to adjust Pharma itself for regenerative medicine, Pharma needs to self-changing and improvement. What value chains in Pharma business will be hit with large impact to adjust to regenerative medicine? That process which got the largest impact is manufacturing. Because especially allogenic cell business, stably producing cell products with the same quality is the key. The next, the research and development and distribution. In addition, so market access and policy issue will be need large innovation. For this process, we need collaboration with many stakeholders, including authority, government, or payers. Okay, and so I just next, so I'd like to talk about what Astellas is doing. Oops. Okay. Yes, to accelerate the development of cell therapy business, Astellas has chosen ophthalmology. Astellas acquired Okata Pharma in 2016. Okata had focused on regenerative medicine and primarily cell therapy for ophthalmology. Okata, with such asset, was the best partner for Astellas. Okata has been renamed as Astellas Institute of Regenerative Medicine, ARM, and so is now an important drug discovery engine of Astellas. The strength of ARM is that it has all the assets of drug discovery research. I mean, the drug discovery research, clinical development, and manufacturing in one organization. So in developing such a state-of-the-art medicines, it is important that these three functions, I mean the research, development, and manufacturing, those three functions work together strongly and unite. This organization makes it possible to carry out cell therapy programs seamlessly and quickly. Here, I'd like to state the importance of utilizing pluripotent stem cells. Pluripotent stem cells can be maintained semi-permanently when proper environment is set. It is also found that differentiation into various cells is achieved by adjusting the culture conditions. From this, pluripotent stem cells can be an important and promising source of regenerative medicine. Astellas has selected pluripotent stem cells as a source of cell therapy that we are using in ophthalmology. ARM is actively developing cell differentiation technology by pluripotent stem cells. As a result of this, it will be possible to produce a large amount of target cells with high quality and purity. Okay, and so I will introduce about Astellas ophthalmology portfolio. This is a picture is illustrated disorder and treatment approaches in cell therapy, showing by the red box in the right side. Astellas has retinal pigment epithelium RP cell programs treating dry age-related macular degeneration, so-called AMD, dry AMD, and staggered macular degeneration as a leading program. In addition, Astellas are investigating the possibility of treating retinitis pigmentosa and macular degeneration by supplementing photoreceptor progenitors, and treating glaucoma and optic neuropathies by supplementing retinal ganglion progenitors. Okay, so here let me introduce the most advanced program of ASP 7317, retinal pigmentosa epithelium, so RPE cells. Retinal pigment epithelium, so RPE cells, have essential roles for normal visual function and survival of retinal cells. In dry AMD, inflammation due to aging occurs in RPE cells. RPE cells disappear, and photoreceptor on the upper part of RPE is lost, and resulting in impaired vision. Therefore, trying to keep this photoreceptor healthy by supplementing this lost RPE is a treatment strategy. We sought to treat dry AMD with RPE cells generated from pluripotent stem cells and transplanting RPE into a subretinal portion. Successfully completed two phase 1-2 clinical trials in the United States using ASP 7317, derived from human ES cells to treat macular degeneration by dry AMD and Stargardt's. So ASP 7317 can be reliably generated from embryonic stem cells. Has studied dozens of the ES cell lines. All reproducible generate RPE cell lines that can be passaged, passaged and characterized and expanded. And it has secure and extensive patent protection of them. Actually, in non-clinical experiments, this is a result of the non-clinical studies. In non-clinical experiments using pathological models rat, RPE was shown to improve visual activity and rescue the photoreceptor. You know, the left side, the bar graph shows us how the visual function recovered, and right fourth panels explains how the photoreceptor maintained. In the clinical trials of phase 1-2, ASP 7317 transplantation showed a positive change on the best correlated visual activity, so-called BCVA of dry AMD and staggered macular degeneration patient. So in addition, this BCVA parameters improvement continues to be sustained 2 or 3 years after transplantation. So in both AMD and Stargardt's patients. Okay, so in the last part, I just touch upon the activity of the Pharma. Pharma is a Japan Forum for Innovative Regenerative Medicine. So Pharma is established 2011. This is a kind of Japanese counterpart of the United States arm. So now 232 Japanese companies joined to Pharma. These are the Pharma's missions. One of the most important work of Pharma is the window activity of industries to authority and overseas organization. So reinforcement of the industrialization is also important for farm. And so also the standardization is also the important activity of farm. So therefore, in the, Dr. Sato's presentation, so he explained the kind of the safety studies, safety studies concept, and so therefore the farm has a close collaboration with his teams. Farm has globally established a relationship with organization or association in several regions, such as with ARM in the United States, CCRM in the North America, and BIA in the United Kingdom, and Sweden and Asian Association. Therefore, when you are planning to establish or develop regenerative medicine and business in Japan, so please contact with Farm. Farm gratefully support you. And every year at Cell and Gene on the Mesa in La Jolla, so Farm has set up a booth and has a luncheon meeting and morning Japanese session, where Farm introduces Japan's advanced approval system and regulations, as well as promising companies. Through such activities, we have strengthened the collaboration with United States regenerative medicine companies. Okay, so finally, let me conclude my presentation. So as I explained previously, regenerative medicine creates new business opportunities, as well as provides novel options of clinical treatment. So this is a very new field and new and promising business field. Next, second, good matching of clinical unmet needs and modalities is an important key to establish regenerative medicine business. Third, value chains in the pharma should be optimized and improved to establish regenerative medicine business. And fourth, Astellas entered regenerative medicine business through ophthalmology segment. The five, pluripotent stem cells can be a powerful modality origin for regenerative medicine. And finally, Forum for Innovative Regenerative Medicine, FARM, is a Japan Industrial Association of Regenerative Medicine, which is reinforcing global collaboration of regenerative medicine. And thank you very much for your attention.
Thank you, Dr. Iwai. Next speaker from Kaneka Corporation, Masato Ibuki. He's assistant manager, Regenerative Medicine and Cell Therapy Laboratories, Kaneka Corporation. Please, Masato.
Good morning. First, I'd like to thank Professor Asada for invitation and kind introduction. Thank you so much. And it's a great honor for me to able to deliver a speech in front of everyone today. I'm Masato Ibuki. I work for Kaneka Corporation and in charge of mass production, in charge of researching and developing technologies for mass production of iPS cells. Today, I would like to talk about a novel tool for suspension culture of iPS cells. In this presentation, please be sure you at least remember to investigate principal repeat controlled size of iPS cell aggregates, and to develop a simple method for the suspension culture of iPS cells. So before starting the presentation about my research work, I would like to introduce Kaneka Corporation briefly. Kaneka was established in 1949 and supply several products, basically chemical products. And we are developing business globally. Of course, the United States is also one of the countries we are promoting main corporate activities. And we manufacture various products in several fields. For example, materials for hair accessories, solar systems, medical devices, supplemental nutrition, and so on. Okay, now let me turn to the main point of my presentation from this slide. For several years, we have started research and development in the field of cell therapy and regenerative medicine. As one of its efforts, we participate in a national project for regenerative medicine to treat diabetes. This slide indicates our current works, and while collaborating with the University of Tokyo, we are developing the mass culture technology of iPS cells. Currently, so many groups actively research to realize regenerative medicine or cell therapy using iPS or ES cell. But in order to spread and infiltrate in the real world and in a real sense, it is dispensable to develop technologies to produce a large number of cells more easily and more cheaply, more efficiently. At the research level, adhesion culture is standard, but its culture scale is limited to make it cost-effective. So now, suspension culture has attracted attention as a mass production method of iPS cells. However, maybe as you know, there are some problems in suspension culture of iPS cells. First, high shear stress induced cell death of iPS cells. So we have to culture gently. And then iPS cells cannot survive alone, so we have to form aggregates in first step of suspension culture. But if aggregates become too huge, they cannot proliferate anymore. That's why we must develop technology to produce efficiently moderate-sized cell aggregates for suspension culture of iPS cells. There are two objectives. First, I would like to talk about investigation of principal lipids regulating size of iPS cells. Previously, our research group discovered that albumin-associated lipid prevented aggregation of iPS cells. But the lipid responsible for this function was not clear, so we searched for the substances that suppress aggregation of iPS cell using aggregation assay. Sorry, figure is too small, but in this assay, a single cell suspension of iPS cell was seeded with candidate lipid and cultured overnight by shaking gently. And then the next day, the lipid improved in inhibition of cell-to-cell adhesion was determined by aggregation size. So now I'll just run through the primary results. When only repeat-free BSA or CDLC was added, huge aggregate was formed. CDLC includes several fatty acids. On the other hand, LPA or S1P was added, small aggregates were formed. So LPA and S1P prevented excessive aggregation of iPS cell. And then we confirmed whether or not it changed depending on the concentration of these repeats. As you can see, these repeats regulated the aggregation size in a dose-dependent manner. So what is essential here is that the aggregation size could be regulated by just adjusting the concentration of this repeat. So I thought that suspension culture is more easily performed even in containers of several sizes and several shapes. So let's move on to the next experiment. We developed a simple method for the suspension culture of iPS cells. We examined expansion culture in suspension using commercial vessels. First, we seeded a single suspension of iPS cell with repeat into 6-well plate and cultured to form aggregate for 1 day and by sinking gently. And then changed medium every day until day 6. And then we passed to 1 liter flask and cultured using same protocol. And finally, we passed to 3 liter flask. Volume was 1.6 liter. As a result, iPS cells increased in suspension using each vessel. Finally, cell number was 18 billion cells per 1.6 liter. And then we confirmed these cells, whether these cells kept pluripotency. We analyzed the pluripotency markers. As shown in these figures, these cells kept pluripotency markers. And then we checked differentiation efficiency to end-down cells. More than 80% differentiated cells expressed SOX17, which was end-down marker. To summarize what I have been talking about, LPA and S1P were the principal repeats regulating aggregation size, and more than billions of iPS cells were produced in simple suspension culture method. In conclusion, LPA and S1P control the aggregation size of iPS cells and enable the simplification of method for the expansion culture system. It means that this is a breakthrough technology to manufacture stem cells cost effectively. Okay. Currently, we are undertaking the development of repeat-based agent for aggregate control. It's a clinical grade, and we will be able to offer it to everyone in the future. And then we continue to develop peripheral technologies and devices such as automatic culture system, and we would like to contribute to the early implementation of regenerative medicine using pluripotent stem cells. Okay, finally, maybe as you have already noticed, my English skill is not so good. Sorry. But our technology is not bad at all. So if you are interested in mass culture system or technologies, please contact us. Thank you for your attention.
Thank you, Masato. Last speaker from Rohto Pharmaceuticals. Her name is Masato Yumoto, and she is a group leader for Basic Research Group, Regenerative Medicine Research and Planning Division for from Rohto Pharmaceuticals. Masato, please. Masayo, please.
Thank you, Dr. Asada and Nakatsuji. And good morning, ladies and gentlemen. I'm Masayo Yumoto from Rohto Pharmaceutical. And it's my great honor to present our latest progress in the development of highly effective cell therapies based on adipose-derived mesenchymal stem cells. Thank you. So before jumping into the main topics, let me begin with a brief introduction of our company. Rohto Pharmaceutical was founded in 1899, about 120 years ago, in Osaka, in Japan. Our initial products were oral digestives, and we soon started to produce eye drops. We have launched hundreds of different kinds of eye drops for various symptoms, and Rohto was recently recognized by the Guinness Book of World Record as a top-selling OTC eye care product brand worldwide. Since the end of the last century, we have expanded our business area into the skincare and diagnosis products. And very, very recently, we entered the regenerative medicine field through cell therapy using mesenchymal stem cells. At Rohto, we use the phrase "never say never" to define our corporate identity. By this, we mean that we will take on a new challenge for a better future to improve the lives of our customers with business activities spanning from food to OTC medicine and beauty to regenerative medicines. Regenerative medicine, of course, is the focus of what I want to talk with you today. As many of you may know, many hurdles still remain in the development of regenerative medicine as a treatment for the wider populations. On one hand, these are biological products which require much more care in handling and storage compared to chemical compound to keep the quality, safety, and efficacy. On the other hand, there are obstacles in the manufacturing process, such as variation in product quality depending on human skills and the ability to scale up to mass production. To overcome these hurdles, Rohto has taken 3 key steps. First, we have developed a chemically defined serum-free medium, which allows us to expand cells in compliance with Japanese regulations. Second, by choosing allogenic transplantation approach, fair amount of cells can be prepared in advance with necessary pre-screening tests and desired functions that exhibit the higher efficacy. And third, through the use of an automated cell culture system, we have been able to decrease the variation in cell quality and the cost derived from manpower. Although we were not the first to enter this area, our company has made significant progress in the development of stable manufacturing techniques in recent years with a company called Micronix, our key partner in automated manufacturing development. Currently, there are two main approaches to regenerative medicine. One is cell tissue replacement, and the other one is cell therapy. As I mentioned earlier, Rohto has made a number of innovations toward the commercialization of a cell therapy approach. The serum-free medium we have developed is optimized for adipose-derived mesenchymal stem cells and complies with biological raw material standards in Japan. And additionally, allogenic transplantation allows us to offer lower cost, pre-screened cell therapy products for a wider range of patients. So let me explain about our first target, liver disease, and the rationale. As shown here, once a liver gets fibrosis, so-called cirrhosis, after inflammation, it could progress into hepatocellular carcinoma. Antiviral drugs for hepatitis C, such as DAA, can be effective for at the early stage of the infection when fibrotic symptoms have not yet developed much. But unfortunately, we currently have no effective drugs for cirrhosis. The mortality rate of the hepatocellular carcinoma is quite high, so we need to stop or at least delay the progress from cirrhosis to hepatocellular carcinoma. But how do we get liver cirrhosis? As shown in the upper schematic diagram, there are two main paths to cirrhosis, one being inflammation upon virus infection, and the other being non-alcoholic fatty liver disease, so-called NAFLD and NASH. In Japan, about 1.5 to 2 million people are infected with hepatitis C virus, and 70% of acute hepatitis C virus infection leads to chronic infection. As for the non-alcoholic fatty liver disease, NAFLD, more than 10 million Japanese are diagnosed every year, and 1.2 million Japanese are diagnosed as NASH. And at this point, there are no effective drugs once the liver reaches the point of cirrhosis. And this fact has driven us to carefully examine anti-fibrotic effect of our MSCs as a potential cell therapy target, cell therapy candidate. We have evaluated our adipose-derived MSCs called ADR001 using several in vitro and in vivo assays for fibrosis. And today, I will show you some of our results obtained from two different mouse models of liver cirrhosis. We first evaluated the ADR001 cells by injecting them into the mouse model of carbon tetrachloride-induced liver fibrosis. Fibrotic area in the liver marked by collagen were clearly smaller in ADR001 injected mouse than that in the vehicle-injected fibrosis mouse. Hydroxychloroquine, an indicative of fibrosis, also was downregulated in the ADR001 mouse, injected mouse, suggesting that the cells we developed have some effects of attenuating the fibrosis reaction. Very importantly, the ADR001 cells also suppressed liver fibrosis in NASH mouse model, as you can see in the right side of our data. With these positive data regarding the efficacy and many other data for safety, which are not shown here today, we moved on to a clinical study. Here's the study design for the clinical study of ADR001. Our collaborator, Professor Shuji Terai in Niigata University, is a principal investigator. He's a clinician scientist in Japan who started autologous cell transplantation for the liver cirrhosis using bone marrow-derived mesenchymal stem cells, and has been working on the development of cell therapy more than 10 years. With Dr. Terai, we started our phase 1/2 clinical trial last summer, targeting patients with a decompensated liver cirrhosis. To evaluate the eligibility of the patients, we use child Pew grading. Patients who have liver cirrhosis caused by chronic hepatitis C or NASH and are 20 years and older and have 7 to 9 points in total after aggregating all the points from the 6 measures shown in the table below are recruited for the clinical study. And I look forward to sharing the results with you next year, hopefully in this meeting. And finally, I will introduce our automated cell culture system. To generate a vast number of cells with a consistent quality for preclinical and clinical study, we developed an automated cell culture system with a closed aseptic features. This system is in compliance with GMP guidelines and has a production capacity of 12 billion cells every 2 weeks. Due to the large-scale production in one lot, we have successfully reduced QA cost and manpower needed. I would now like to play for you a short video that gives more details on our automated cell culture system.
One of the keys to using stem cells for therapy is to produce a large number of cells of good quality at high reproducibility. Automation of cell culture processes enables us to produce cells fulfilling the above-mentioned requirements. We are currently developing such an automated cell manufacturing system in which adipose-derived MSCs are maintained, expanded, and formulated for use in clinical applications. The automated cell manufacturing system consists of an isolated cell culture unit, compatible CO2 incubators, and a cell storage unit. The cell culture unit can connect with the incubators and the storage unit in an aseptic manner. In the cell culture unit, the isolator, there are a microscope to monitor culturing cells, a centrifuge to spin down cells, and last but not least, 5 multi-axis robotic arms to perform multiple tasks necessary for cell culture. Our automated cell manufacturing system does not require human intervention. Thus, it secures aseptic conditions, minimizes batch-to-batch variability, and reduces cost and labor for production. The tubes are then transferred to a storage unit, frozen, and stored in a liquid nitrogen tank. Taken together, we believe that our automation system is beneficial to patients who are waiting for stem cell therapy to be developed.
I hope you enjoyed the video. And the system can be reconfigured to the culture of various cell types other than MSCs. But we are currently focusing on the preparation of MSCs. As shown in this table, we carefully examine the characteristics of MSCs in several parameters such as cell surface markers, growth factor secretion, and the growth potential, which is shown as delta PDL here, and capacity of Tori lineage differentiation. The results show that our automated system is slightly better than manual preparation, which means we can now pass the time-consuming and demanding cell culture work onto our very talented and hardworking machines. With the development of the automated system, we have gained the capacity for mass production and reduced cost. And more importantly, we believe that this system allows us to produce consistently high-quality products by eliminating the variation derived from human skill. Looking toward the future, we will further advance our mission of implementing effective cell therapies. We think our research and development has big potential, and we plan to expand the indications toward currently incurable disease like pulmonary fibrosis, heart failure, neurodegenerative disease, and inflammatory bowel disease after obtaining the approval for liver cirrhosis. Thank you.
Thank you, Masayo. We have finished all the speakers, and we have several time left. Is there any questions for all the speakers to raise hand? Yes, please. Please microphone to the.
Yeah, first, thank you. I'm Susan Sang from Kansas State University. I have a couple questions. One is about iPSC. Targeted differentiation, what is the purity, and also the targeted differentiation quality criteria? And that is one thing. And then another question is for the last speaker, for your MSC production, is that on the two-dimensional surface production? And how that the variations from the original primary MSC, and then how that would, for clinical uses, and then what is the risk?
Right. For the first question, who want to talk to for the speakers? The first speaker に先生答えますか。じゃあ。
I presume the second question is about MSC, maybe. But first one is differentiation efficiency from the iPSC. So, yeah, it depends on different among the who is doing what differentiation to what cell types. Okay, so if your question is specific to someone's differentiation, please tell that. But for myself, I'm particularly focused on the practical application by differentiation. So basic scientists, biologists start to use rather natural differentiation inducing cytokines. So in our body, cytokines is doing that job. But cytokines is very difficult for mass production with high quality and low cost. So we try to make use of the small chemical compound to replace the cytokine. In that way, cost is much lower and reliability much higher. So for cardiomyocyte, at least we succeeded to induce cardiomyocyte from ES or iPS cells using only small chemical compound without any cytokines, even without any proteins. So that's the ideal system for the application.
Please, second one.
Thank you for the question. So our current system is 2D cell culture system with plastic. And so your another question was probably the variation after culturing on the cell surface, right? And in the first, passage 1 or 2, we have several, different type of cells. But after culturing with our media, a certain population was enriched, and it has a pretty much pure population as far as we checked with our cell surface markers, like CD90, 105, 73, or 34 or 45, which is negative markers. So we believe that it's pretty much homogeneous.
Okay, thank you. Is there any questions?
Oh, regarding the first question.
Oh, okay, right.
So regarding the first question, the less is the amount of hazardous cells like residual iPS cell or malignant transform cells, the better is the quality of the product, cell therapy product. So, but the problem is that we don't know well about the sensitivity of the test method for the detection of the hazardous cells. So that's why we are trying to have a consensus about the characteristics of test method about tumorigenicity, tumorigenic cells.
Right. Okay. Any other questions? No? Okay. Yes. Yeah. Please use the microphone to wait for 4 minutes. Thank you.
So, my question is about the scaffold you use for your the myocyte regeneration. So you use aligned nylon fiber, right? So first is what's the degradation rate? So how long it will exist before?
So,
Yeah, there are many kinds of scaffold used by many groups. So you could find some review article maybe, but for ourselves, we are using so-called nanofiber that's produced by electro spinning. So that's use a polymer solution, and by charge difference, so that makes you can control the thickness so the degradation rate or other specifics can be controlled by the composition of the solution mixture. So, and we could use, for example, very stable fibers by using something like polystyrene or something. But that for the device production purpose. So it's fiber lasts long term. But for the transplantation study or other study, so there are several kinds of polymers which has different rate of degradation, and we can choose that. So it's depends on something. There are some fiber in the which degrade in several days, for example, short, or last 1 month. So there are many kinds of technology polymers. So you can choose among that.
Right. Thank you. Thank you for attending this Japan Symposium. We have will be next year again to get some same Japan Symposium here. Thank you very much for your audience. Thank you very much.